All right, I think we can get started since we're right at the top of the hour. My name is Matt Van Hook. I'm at the University of Nebraska Medical Center, and I'm the chair of the RESTORE Virtual Discussion Committee. So one of the things that we try and do in our effort to push forward the frontiers of vision restoration and optic neuropathies is hold these virtual discussions. Our goal is to hold these about 3 times per year, and we're kind of, right around that time frame. But really, these are opportunities to hear from experts on some of the kind of most pressing edge of the frontier topics as it relates to vision restoration. So we've had some on sort of clinical translational process and hurdles. And some other topics. And if you're interested in looking at those, you can find those up on the RESTORE website. Um, our topic today is looking at axon growth and synaptogenesis in the retina and retinal projections in the brain. Oh, sorry, and don't be offended if I come and preemptively mute you, just because I'm trying to keep excess audio from leaking in. So we have a really nice set of speakers today who are providing some overview of these topics. I'm going to have them introduce themselves in just a minute. The way that we structure these is that we've invited a couple of speakers with some expertise to share from their experience and share some perspectives. And then really our goal is to open up the floor for as much discussion as possible. So our facilitators are here to help that discussion go smoothly. And then it really comes down to all of you to chime in with your ideas and thoughts and questions for the group, for each other. It's not a simply asking our presenters and having them respond. It's the sort of thing where anyone can ask a question and anyone who's participating in the discussion can chime in. So my goal here is to mostly stay out of the way and let this be a smooth discussion. Try and make sure that we get a chance to address some of the pressing topics. So again, thank you for joining, and I hope you get a lot out of this. So I want our speakers to introduce themselves first. So Elizabeth, I'm going to turn it over to you and have you just tell us very quickly about yourself, your affiliation, and your background.
Okay, so hello everyone. My name is Elizabeth Zuniga-Sanchez. I'm an assistant professor in the Department of Ophthalmology, and I have a joint appointment in the Department of Neuroscience at Baylor College of Medicine. And I study, I'm largely interested in neural circuit formation using the mouse retina as a model system.
Great. Greg, how about you introduce yourself?
Great. I'm Greg Schwartz. I'm an associate professor in ophthalmology and neuroscience at Northwestern, Feinberg School of Medicine. My lab mostly works on retinal computation inside the retina, increasingly to the brain and behavior as well. Bunch of computational stuff. I'm not really in the vision restoration field, but I am here to kind of talk about physiology and function of ganglion cells.
Wonderful. Awesome. Supraja.
Hi, everyone. I'm Supraja Varadarajan, and I'm an assistant professor. Recently started my lab at UT Southwestern with an appointment in neuroscience and a secondary in ophthalmology. And my lab is interested in axon regeneration of the visual system, particularly in the mechanisms of underlying activity-induced repair.
Wonderful. Thank you. All right. So I'm going to stop sharing my title slide here. And Elizabeth, if you want to start sharing yours and take it away with your topic.
Okay, can everyone see my screen? Yes? Okay, perfect.
Yes.
All right, so I'll go ahead and get started. All right. So synaptic connectivity in the mammalian retina. This is a very simplistic overview of the retina circuit that you have photoreceptors, your rods and cones that form synapses with your inner neurons. These then make synapses to your retinal ganglion cells. And so for this RESTORE meeting, we've been really interested in what happens when you lose these retinal ganglion cells. Not only what happens when you lose them, but how do we bring them back and how do we have them restore their connections? And so this is how I kind of see it, that you have these very different retinal ganglion cell, and they're all different subtypes, and they all express very unique set of genes. And the idea is that you have the postsynaptic neurons that then need to once again form connections to these presynaptic neurons, but that this is a highly selective mechanism where the precise partners of each one needs to find each other again and make the appropriate connections. So very simplistic view, but that's kind of how I wanted to display it. And so the key question that this leads me is to what are the key molecules, or what is the key, you can imagine, building block that you need to restore these connections. And so I actually don't study the inner retina, I actually study the outer retina. But that's what my whole presentation is about, is like, can we gain information or insights into this mechanism of restoring connectivity by looking at what we already know on the other side, which is in the photoreceptors. So something that I really like, or why I'm intrigued to studying the photoreceptor connectivity, is that if you look at, or you stain the different neurons or retinal neurons, here we're staining the horizontal cells and the rod bipolars, they send out their processes and they're confined to this region, this particular region called the outer plexiform layer, which is the synaptic layer. And so this also, the axon terminals of photoreceptors are also restricted to this area, and you only have synapses in this particular location. So then when you have any type of wiring defects, such as the case here of losing beta-2 spectrin, you see now that there's misprojections of these processes where they're extending beyond the synaptic layer. And so you can easily visualize when you have a connectivity defect, especially in the outer retina. We also have a lot of different tools or different antibodies. For example, here I'm showing mGluR6 staining in one of a published paper. And here they were able to show that in the LFN1 knockout, when you stain with mGluR6, you lose the staining in the rod synapses, but the cone synapses, it's still retained. And then you can further use high resolution imaging techniques, this case transmission electron microscopy, and see that the dendrites of rod bipolar showed here in red fail to invaginate and form synapses at these rods, which confirms the findings in the confocals. And then also in the outer retina, we have different functional in vivo tests that we can use. This one is ERGs. We can measure responses from the photoreceptors. We can measure their downstream bipolar responses as well. So we have all these different techniques that allows us to screen for wiring defects in the outer retina. Now, using these techniques, I'm trying to summarize, many, many years of field of literature, but these studies have uncovered, you have different molecules at the synapse. You have ribbon and active zone proteins. You have synaptic vesicle proteins. And there's a slew of cell adhesion molecule, extracellular matrix proteins, and postsynaptic receptors that are all expressed at these photoreceptor synapses. So there's multiple molecules here. And then recently, with single cell sequencing, we noticed that now we can identify the set of genes that are differentially expressed between different cell types. So even within photoreceptors, you have the rods and the cones. We notice that they express unique set of genes in the adult retina. And this lends to why they, in terms of their synapses, they look very different from each other. They have different composition and they have different postsynaptic targets. So even though they're photoreceptors, each unique photoreceptors has a unique set of genes that allows them to make their specific connections. And it's really been this boom of identifying these different molecules that are expressed in distinct subtypes and then lead us to identify defects in particular pathways. So there's been multiple cell adhesion molecules that are restricted to a particular cell type, such as a cone or a rod. And when we knock them out, we notice that we have particular defects in that particular pathway and not the other. So the idea that you have these cell type specific synaptic determinants in these different cell types. And so not only do we have cell type specific molecules, but as a developmental biologist, realize there are developmental mechanisms important in retinal circuit formation. And to highlight this, I wanted to bring the example of the horizontal cell. So the horizontal cell is an interneuron. And what's amazing about it is that the dendrites synapse selectively to cone photoreceptor, while the axon terminal synapses to the rod photoreceptors. This is in the adult mouse retina. Oops. But then during development, there's been, several labs have shown that very early on, horizontal cells extend out neurites around postnatal day 3, and these make contacts to cone photoreceptors. A few days later, they extend out an axon, and until around P10 is when the axon terminal extends out their processes and makes synapses to rod photoreceptors. So this tells us that connectivity or horizontal cell connectivity to these photoreceptors are occurring in a stepwise manner across development. So it doesn't happen at the same time, but there's particular order in which this happens. And so the same way that we think that there's connectivity that happens in the stepwise manners, we profiled these horizontal cells and found that indeed they're expressing different set of genes across these different time points at these very early time points. And then if we do pseudo time analysis, we find that there are certain genes that they're upregulating, downregulating that may be what's responsible for their precise connectivity with cones first, and then followed by those of rod photoreceptors. So the idea is that wiring molecules or these molecules needed to restore connections, they might not be present only at one time points, but we need to follow them across development when they're making these connections to their appropriate partners. Okay, so I just talk about neuron to neuron interactions, the presynaptic, postsynaptic neurons, but then I also want to highlight that that's not the only thing in the retina. And so the ganglion cells, as well to other retinal cell types, they also make contacts to other cells. And so the idea is we can't disregard these. There's other non-neuronal cells like the glia support, the Mueller glia, the astrocytes, and even the vasculature that these neurons need to be able to connect once again and be able to find or receive the nutrients required for proper survival and support. And so the take-home message behind this is that when we think about restoring these connections between ganglion cells and knowing that there are very different subtypes out there, we also have to think about when we think about which are the key molecules to restore these, we have to acknowledge that there's cell type specific expression, that there's developmental time expression. So when and where this molecule is required, we have to consider. 2, we can't rule out the connectivity to other cell types. This is non-neuronal ones, that they also have to receive the proper support from the vasculature, from other glia for their integration and survival. And then thinking again how the tools that we have for the outer retina, thinking once again how in the inner retina we can use something similar to assess when we have proper rewiring and a proper restoration of vision. So I'll stop there.
Wonderful. Thanks. Greg, go ahead.
Hey, thank you so much. Let me share my screen here. Okay, you guys can see that, right?
Yes.
Okay. Thanks for inviting me here, and it's first of all just wonderful to see in this Zoom room a lot of friends and folks I haven't seen for a while, and it's great to see many people here. I want to take a little bit of a different approach given the expertise of everybody that's here. I don't want to try to teach you a bunch of things about ganglion cells or all the different types. I'm happy to discuss that. I want it to be a discussion, but I want to try to take an honest take of where we are in this field and what we're actually trying to do. And I mean, I'm going to do it with some strong words, but also I'm going to try to be humble about the fact that this isn't my field, and please correct me and like let's have a debate. That's what we should be doing here. But what are we really doing here? Like, what are we restoring? Are we trying to have transplantation, real functional restoration, and axon regrowth? If the point is to try to help people see, we need all of those things. And why and in what diseases are we really doing this? So this question that's on this slide was from my RReSTORe intro in 2022 when this thing was getting started. And I want to know how far we've come since then. We know that there's a huge amount that we don't know about dendritogenesis of ganglion cells, synaptogenesis, to get successful integration, proper connectivity into the inner retinal circuit, or at least some kind of connectivity that's lasting and that can give you actual functional signals down the optic nerve. And then how is any of this going to work following transplantation or doing something to save the cells that are still there? So one of the things that Matt asked us to talk about is how do we measure success? And I think we have to not fool ourselves. I think we need actual measurements and goalposts. And I want to know where we've gotten with any of these. So following transplantation or following anything to try to restore vision, where are the goalposts and where do we actually need to get to restore vision to a patient? We need RGCs to extend dendrites into the IPL, right? Transplanted ones or the ones that remain, they need to extend some dendrites for sure. And all the way at the other end, we want synaptic connectivity that's like indistinguishable from normally established for every ganglion cell type, right? That's like the goal all the way on the other side. Maybe we don't need it for every ganglion cell type, and I'll talk about that in a moment. But where are all the measurements in between? Expression, I'm going to list a few that I've thought of, but I'm sure this is not a complete list. Expression of functional glutamate receptors, program specificity for the inner versus the outer IPL, program specificity for particular bipolar connections, also to glial cells and everything, connections to different amacrine cells that shape receptive field properties and temporal profiles. And then part of the question is, can visual experience push us downfield onto these goalposts and how far? And there's a whole bunch of other things I could list as well. Intrinsic properties of the ganglion cells being fairly normal, them not dying because of the pathological condition that they're in the first place because of the disease, metabolic regulation. And it's just, we could list for a long time all the things we don't know about this, but has anyone recorded a light response of any kind from a transplanted ganglion cell in 2025? Electrophysiologically? Like, I think the answer is no, right? So, like, there's a lot of details we can talk about not getting right, but like, we're not close if we don't have any synaptic integration of these cells yet, right? So, how are some of the ways we can reach this? Sure, there's a lot of single cell RNA-seq we can do and learn about the molecules that are important in this. And I think a lot of you, this is not my expertise, a lot of you are doing this. There's a lot to do here. This is molecular engineering of transplanted ganglion cells, stem cells, and organoids. I know we have experts here to try to program the cells. There's characterizations of these transplanted ganglion cells, but I'd urge people not to just do microscopy, but to also do electrophysiology on them and see if they even have any light responses or correct intrinsic properties at all. And then ultimately, one of the gorillas in the room here is how much do we need to do this for all the different cell types? I love studying ganglion cell types. I've, committed many years of my life to figuring out all the different ganglion cell types in the mouse retina and where they go and what they do. But does it really matter if we're trying to restore vision in people? if one of the 40 or so types in the human that does something we don't even know in peripheral retina is correctly connected or not? Probably not. Like, let's get the midgets and parasols right. Period. Like, really, who cares about the specificity? Or even if you just got the midgets right, like, who cares about the specificity of the connections of all those other types or anything? Can we get cells that have on and off light responses at all integrated into a circuit that then extends an axon somewhere in the brain that talks to something? Right? Like, that's kind of where we need to get first before we worry about all the details of all the different types and everything. So I think, I want to have this open debate about this. I want to discuss it, but I think we need to be realistic about where we are and that we need to test things with electrophysiology, not just with the cells being alive and some microscopy that suggests that they might be sort of in the right place. But do any of these things have light responses? And can we do that? So yeah, I'll leave it to that, and I'll leave it to the next speaker to talk about the next part about the axons actually making it into the brain. But within the retina itself, I think we have a lot of work to do to get light responses and getting themselves first.
All right. Thanks, Greg. Supraja, you want to share your slides and take it away?
All right, can everyone see this?
Yes.
Okay, great. All right, so my absolute favorite topic is axon growth in health and disease. And so I'm really excited to facilitate the discussion on the challenges we're facing in retinal connectivity or reconnectivity after injury and disease. And I'd like to take a sort of simplistic view and compartmentalize the milestones that we need to achieve for repair. There's been a lot of discussion, especially in the previous RESTORE discussions, about cell survival and transplantation and neuroprotection. But today we're focusing on regeneration, axon regeneration, and reconnectivity. And really, I think after the last many decades of work, the big challenge that we're facing right now is how do we get these regenerating axons to grow past the chiasm and into the brain and in high enough numbers. And so is there something that we can do to perhaps get them to grow past the chiasm? And maybe that will also facilitate higher numbers of axons regenerating into the brain. And as far as reconnectivity goes, we're really thinking about how do these axons identify where to connect to, how they become remyelinated, and how do we restore image forming and non-image forming visual functions. And I think the main point that I kind of want to hone in on is looking at the optic chiasm, which happens to be a major navigational choice point, and looking at what lessons we can learn from recent findings about leveraging guidance cues, neural activity, and electrical stimulation. And so as we know, we've had a lot of success in getting axons to regenerate down the optic nerve, and kind of what this picture is trying to depict. We can get them to the chiasm, but we know that there's lots of errors in what they do beyond that, or even getting them past that chiasm, and not enough of them make it into the brain. So a few studies that I want to highlight today, and again, like Elizabeth said, it's impossible to summarize all the really cool studies, so I focused on a particular few ones. But in this first study from Kim Gokowski's lab, there's a really cool approach to using electrical stimulation, where you're stimulating the optic nerve using rebalancing the charge, if I'm saying that correct. I think Kim is there to help us through this. But basically, in trying different types of waveforms, they've shown that there is a particular waveform that you can stimulate the optic nerve in rats and get a really robust regeneration, as seen in this last panel. And I think it's really cool because we've looked at many molecular mechanisms, many different types of stimulation, but over here we're using electrodes to really stimulate the ends of the optic nerve and get more growth to the chiasm. And another cool study from Hawaii's lab has shown, kind of also focused on how do we get them into and past the chiasm, where we know again that this problem exists where many axons that even manage to regenerate end up rerouted into the opposite optic nerve and really don't make it past that point. And so there aren't enough getting into the brain. And not this paper that I'm showing you today, but in another paper that they came out with, it kind of sets up the landscape of all the guidance signals in the adult brain before and after injury, which is very helpful to kind of get a sense of what is in the brain and what can we leverage or silence or and enhance. But in this particular study, they show that the slit-robo signals are particularly important, especially at the SCN, which is one of the first targets that these axons innervate in the brain. And by silencing robo 1 and 2 conditionally, you can get more axons to re-innervate the SCN. And I believe a part of these are also the IPRGCs, so there's some specificity to that process that's happening just by silencing some of these repulsive signals. And of course, we know from a whole body of work from the Mason lab and others that the optic chiasm is really, like I said, it's a very tricky navigational choice point, which is where many axons get misrouted, both in development as well as in regeneration. And there are many different signals present there, a combination of adhesive, repulsive, attractive signals. And these studies on the right also talk about how several chemokines can be involved in guiding these RGC axons, either by silencing them or leveraging those ligand receptor interactions. And so all of these are really important lessons to think about. How do we use some of these either together or in combination with other approaches to get axons past the chiasm and into the brain in higher numbers? But of course, the counterpart to this is using slightly different models and moving a little bit away from the optic nerve crush to get a sense of other things that might be happening in the repair scenario. So this paper from Shigang He's lab talks about how they move the injury more distal and lesion the optic tract in the brain, and they really kind of show us that these axons don't spontaneously remyelinate in adults, right? They don't regenerate very well, and if we get them to regenerate, they're not really remyelinating very well. So that's probably also contributing to why we see such less functional restoration. And this other paper from Mikhail Granato's lab highlights this very important and interesting finding, because they use larval zebrafish. But they show that if you completely induce an optic nerve transaction, these axons happen to innervate both the optic tectums in both hemispheres of the brain, which doesn't happen normally. But if you induce a partial lesion, a partial optic transection to varying degrees of severity, then somehow those paired axons are helpful in getting those regenerating axons to the correct optic tectum, which I thought was really cool, because that again highlights to the different types of signals that we haven't really begun to start considering, right? These are important cues. Axon fasciculation signals are very important during development. You always have a pioneering axon that explores more, and the following axons that explore less and have a paved pathway or a scaffold to grow along the pioneering axons. So this somehow hits at those principles. And another cool study from Lief Moon's lab has shown how optogenetically stimulating the superior colliculus in a mouse model of glaucoma can confer neuroprotective properties on the retinal ganglion cells. So this is now looking at how do we leverage other signals and stimulate retinal recipient targets. And so in my previous lab, my postdoc work from the Huberman lab has kind of leveraged all of these and said, okay, if we put all of these together and take this partial transaction where we basically induce a partial optic tract transaction in the brain and chemogenetically stimulate the retinal recipient neurons in the pretectum, we see that we can promote robust regeneration. And so to very quickly summarize those results, we basically see robust regeneration that can get past the lesion site as well as the stimulation site up to 2 millimeters in 2 weeks. They re-innervate both the minor targets of the pretectum as well as the major target in the superior colliculus, and they rescue some optomotor deficits. And so I think all of these together are highlighting different injury models that we can begin to also incorporate, particularly the partial injury models that a few of us have been using can be useful to mimic certain aspects of diseases as opposed to a complete uniform lesion. And it also, and we've shown in our own hands that whether we stimulate neurons broadly distal to the lesion or selectively in a specific retina recipient target, all of these are sufficient to promote regeneration. And other work from Matt, Josh Morgan, and Pete Williams have, and I think this was also covered in a previous discussion, where they've started looking at what is actually happening in the retinal recipient target nuclei themselves, both in terms of the target neurons, but also in terms of the synapses between the RGC axons and the target neurons. And how are all of those impacted in different models of injury or disease, which is an important thing to consider, because I think we've talked about how one point is that even if we can get these axons to regenerate, we need something there to receive them. And so an equally important consideration is what is happening to the retinal recipient target neurons, and how do we leverage those signals from the brain, just as has been shown it is very important during development. How do we kind of start integrating all of these signals? So the points that I'm putting forth for discussion today is how do we specify signals all along the retinofugal pathway at the lesion, distal to the lesion, at the chiasm or the targets. And this could be in many different ways of modulating different signaling pathways, guidance cues, either electrical stimulation or neural activity, or even other mechanisms. But really, I'm very excited about how this group can come together and start integrating signals from both the retina as well as the brain to really leverage that, because that's what these axons do during development. They're not meant to receive signals from one point and keep going forth to their targets. They have molecular signals all along their trajectory that shape their trajectory very intentionally. So how do we kind of integrate everything that we've learned so far into specifying signals all along the retinofugal circuits? And I think one important consideration that Elizabeth also brought up is considering non-neuronal cells, right? Like glial cells are very important. At the chiasm, the radial glia are some of the cells that specify these guidance cues. Biomaterials have been used in spinal cord injuries to express some of these molecular cues, and how can we leverage that here? And also learning lessons from spared axon escalation signals on promoting sufficient regeneration. But also to hit on another note of what is the different measures of success is how do we move past OMR, right? Just the optomotor reflex. The OMR is a fantastic way to get at a starting point of do we see any functional restoration, but it is a very broad behavior. It's a reflex. And so how do we start using or identifying other specific visual behaviors to really gauge at recovery and specificity? And I think I will stop with that.
Wonderful. Thank you all. All right, so what I'd like to do is kind of open the floor for discussion here. There was some stuff that came up in the chat, so that might be a good place to start, and we'll sort of see where things develop from there.
Yeah, I mean we can yeah, so again, please try don't take offense to this. I want this to be a spirited debate, and I love that people who knew more about what's going on in this field are posting things. Thanks Jonathan for posting that paper with the potential calcium responses. Yeah, I'm not convinced. I don't know, I'm just looking at this right now. That looks like a calcium oscillation that's 20 seconds long that has nothing to do with light necessarily. But I don't know. I mean, you're right. And then Brad posted that Ben Severe and Joe Leffler are actually testing transplanted ganglion cells in macaque and looking for synaptic currents, which sounds like the right thing, right? I mean, the most direct thing to do to see if there's any possible light response. And so far, not seeing anything, but that seems like, yeah, the most direct test. So,
Greg, I think after you've had a chance to read the paper, I know you just got
yeah.
glance at it.
Yeah.
It's calcium response is stimulated by light.
Okay.
They don't just sit there and happen every 20 seconds. So there definitely is something going on. Now, I think you're asking the question of how much can we further challenge the system to show that these are electrical responses that behave in a ganglion cell.
I think he showed also they're blocked by strychnine, which is another little piece of that puzzle evidence anyway.
That's interesting. They're blocked by strict. Okay. So they're inhibitory somehow? Like there's an inhibitory circuit that's driving them or something? I don't know. Yeah, no, I mean I definitely need to read this paper then carefully and see what's going on, but it's just calcium
I
is
don't know if
not.
that's in the paper. I've seen it. I
Yeah.
think I've seen it at, on the screen at a conference.
I don't, right. I'm sure. Yeah, light can do a lot of things in the retina. There can be a lot of connections to the time course of them looks, yeah, sure. They look triggered by light. They're very long lasting, but yeah, I mean that's getting there.
That's interesting. The time course, Hiroshiga in Adriano Dipolo's lab just before he started his own lab is a paper demonstrating that injured RGCs in the context of experimental glaucoma in mice have a very long, sluggish tail on their calcium responses.
Yeah.
The recovery is very prolonged.
Yeah, I'm always wary of certainly calcium responses are a great place to start to look at these things, of course, but there's so many things that can drive calcium, metabolic damage, all sorts of stuff. Like I've been just doing a bunch of that in my lab, and its relation to actual spikes, especially in a non-native, like potentially damaged cell, can be really indirect. So it's fine. It's a great start. It's just, yeah, until I see spikes driving that are actually going down the optic nerve.
Yeah, I know for myself, sort of being a patch clamp electrophysiologist, being able to actually patch a neuron, listen to what it's saying in real time, sort of on that really high temporal resolution, is something that makes me kind of more convinced. I can really get a look and a see into what the neuron is saying. So that's
Right.
kind of this really good standard of evidence in my mind in terms of an experimental system. And so, I think looking for the, EPSCs, EPSP or IPSPs in, transplanted neurons, I think is, beautiful and the sort of thing that's needed.
Yeah. And I think we also have to be honest about that we're transplanting them into a damaged system, right? I
This
mean,
concerns me about growing an axon up the sick optic nerve. Aren't we just going to kill it all over again? You have
yeah,
to fix
yeah.
glaucoma in the first place,
Right,
in order to
exactly.
restore vision successfully. Yeah.
Right. I think that's right. Brad points out right that that's true in the optic nerve. There could be scars there, but it's also true separately in the retina, depending on which disease you're talking about, right? I mean, you could have abnormalities in the retina, both from glaucoma, pressure-related things, or anything related to that, but also Rich Kramer's work on AMD and other diseases that affect photoreceptors that show that retinoic acid then can then alter the intrinsic properties of the ganglion cells that are left. So photoreceptor degenerations, even though they have nothing technically to do with the ganglion cells, can then alter their synaptic properties, make them fire a bunch more, and even the ones that grew up there are having trouble. So yeah, it feels like we're a long way from restoring useful vision by transplanting stuff. But
I mean, with regards to regrowing an axon and the cues that are necessary for that to even happen, I'm curious, Elizabeth, you were alluding to a single cell data set that you had across different postnatal days for the horizontal axon extension. Did you detect any interesting cues that you thought are in any way similar to what happens maybe during axon projection of RGCs? Like, is the recipe for extending an axon the same across neuronal cell types? And what was sort of the discovery there that could help us?
Yeah, thank you, Marina. So actually, we found a lot of robo slits, which Supraja when she mentioned that data, and I was like, it makes sense that those would be likely candidates that, like you alluded, that you have this general mechanism of the axon needs to grow out, and they're sensing these cues across it. So I was thinking we have not like try to restore or the native form of ganglion cells, but kind of like this engineered neuron that, like you mentioned, has the signals to extend an axon and find its way. Um, because during development, there's all these unique sets.
Yeah, and I think this was already brought up in previous RReSTORe discussions. How faithful do we have to be to actual development? Like you're saying, maybe just getting an engineered cell that does the job we want it to is more than enough. It doesn't have to be a specific RGC subtype. It doesn't need to have the exact identity. It might even not have to connect to the partners that the endogenous RGC subtype would normally. Maybe it's enough to just create the connections between, and a bipolar cell and a meaningful target, and that can still do the trick. I mean, we know this from the optogenetic field, just adding a channel rhodopsin to, a bipolar cell might still evoke some sort of meaningful response that can be decoded somehow. But yeah, I'd love to hear more about or see. Is there a paper that you've recently put out with all of these different markers?
We have it in revision, so hopefully it will be out soon.
If there's a
But Seth
paper in
Blackshaw,
Lab,
he has some beautiful like developmental data from Brian Clark when he did his paper, and we're seeing kind of very similar molecules that, depending when you sequence, you see the upregulation, downregulation of a lot of these molecules. And like you mentioned, some of them, we know that like efferent signaling, roboslits, they're involved in this early axon guidance mechanisms. Yeah.
I'd like to propose a thought, and we can bounce it around a little bit. And that is actually two parts. One is maybe we don't need two eyes. So one eye is great for people when they don't have any vision. And then the second is maybe we just need to get the ganglion cells to follow the remaining ganglion cells in that eye, because usually with glaucoma or any of these diseases, optic neuropathies, you don't lose all the ganglion cell axons. You lose a lot of the ganglion cell axons, but if they just follow the ones that are there, and you don't have to worry so much about the chiasm, because the ones that get to the right side, we'll take them, and the other guys, we can figure out a way to get rid of them. But, as long as some get through to the right, cross at the chiasm, get to the right side of the LGN, of the brain, so that they can innervate the LGN, I think that's almost all you really need to get, as long as you can get a map. But to me, I think the thing that is maybe the biggest hurdle is getting the map to form in the LGN, because you could probably get some axons back there. It's looking like better and better. We're getting some. As long as they just track the existing ones, great, they'll get to the right target. And then if we can get a map, it's going to be as good as a prosthetic today anyway, as good as a chip will be for at least a while. So I don't think you need to get that many of these axons back to the right target, but you do need a map. And I think it's interesting to think back on the Xenopus days of, axon guidance and map formation in the tectum, and fish. The evidence from there is they retain those cues in those animals for correct mapping. And I don't know if anybody has any, information about whether we still have, it sounds like, from these partial lesions of the tract, that Supraja was talking about, that, the mapping function is still there for ganglion cell axons if you can get them past those lesions. So maybe it's not, we don't need to re-engineer the whole pathway, is what I would say. If we can save ourselves having to do that, I think there's a shot. Re-engineering the whole pathway, yeah, probably never going to happen. So anyway, I'd like to hear people's thoughts on that.
Just a question on that. So, that certainly happens during development where axon, there's like kind of a pioneer axon and then the other axons will follow along. But wouldn't that be a challenge after they're myelinated, like they're, kind of covered by these oligodendrocytes? So would the signal have to be like the oligodendrocyte itself instead of the axon?
There's a lot of unmyelinated axons in the nerve. I don't know how many of them go to the LGN versus other places, but yeah, I think you raise a good point. We know the nerve is already not a great environment for growth, partially because of inhibitory factors from oligos and from myelin. It's pretty clear, but that doesn't seem to stop them when you use the PTN, the PTEN mutations and things like that. That does seem like they can still grow along that pathway. So
Right.
again, it's probably not ideal for them.
Or maybe it could be like if you could get a few to grow, then the others could kind of follow along with it. And they'd be, regenerating ones that are following the same tract, something like that. Yeah. It's a great thought though.
No, I like that idea too. I mean, I think that's something that we don't know as well, right? Like how much of this is how many of these axons get demyelinated, and is that necessary for those remaining axons to guide the ones that are trying to repair themselves? We don't know any of that, and I think that's all of that is just a big black box, in terms of both during development, but also in repair, right? And I think one thing that's remarkable in all of these partial transactions is that they, like Tom said too, they don't really stray out of the optic tract, and they don't really innervate any other regions other than visual target nuclei. Whether these different subtypes are now innervating different targets, we don't know. We don't know whether that we care about that, right? Like we want to get some axons into some targets, and then we'll figure out what they're restoring to begin with. But I think keeping in mind that, I think these are really important points on how do we turn on those signals, in anything that is remaining.
So two points to that, and maybe it's not as relevant to transplant, but in the context of optic nerve transection in a highly regenerative species like zebrafish, the axons aren't always going to the right location. We see quite a bit of aberrant growth in that context, and we've begun to phenotype them according to their essentially regenerative capacity, and then try to do the omics on that to get a handle on what's going right and what's going wrong. But secondarily, to Nick's point about the pioneering axons, another trick we've been up to is placing human RGCs into the zebrafish retina and asking what their intrinsic regrowth capacity is. And in that context, we've noticed if we ablate the endogenous RGCs such that they're naturally regenerating, the human RGCs seem to have a better capacity to grow out of the eye. So if they have a leader, apparently all can go, well. So I guess that brings up the point of maybe transplanting zebrafish RGCs into the human eye with the human RGCs to give them the leaders, I guess, the pioneers. So yeah, one goofy idea.
Yeah, just on that point about these regenerating species, I did my postdoc working on frog regeneration in Rana pipiens, and it's pretty spectacular how well they can grow back to the tectum. But as you say, Martha Patton used to make these three-eyed frogs, and when we looked at those, the extra eye would just literally grow, over the tectum and then innervate because it was sitting in a completely ectopic spot. So those axons didn't need to follow the normal trajectory. Even though there was beautiful order within the optic nerve and optic tract itself, it didn't need that order to find the right target sites. So back in that era, there was debate over whether the order in the optic nerve and tract were necessary for giving rise to the appropriate connectivity when they get to their targets. And these are redundant mechanisms. And as you say, they make a lot of mistakes. Axons will grow back up the wrong, the contralateral optic nerve back to the eye. In some cases, you'll see these really big problems. But in the end, they see really well after having regenerated their optic nerves. So the connectivity and the size of the terminal arbors in the tectum, all these things just reorder and resort. So that map in the tectum and presumably the LGN for us would be, one of the key things that we have to, make sure the axons get right if we're going to get, useful vision, because it's got to be a map. If it pretty much has to be a map.
That's a really good point. I should have made that. They make mistakes, but once they get to the tectum, they home in. That's an excellent point. They can wander for days, but as long as they find that tectum, then they calm down and innervate.
So I see, Art, in the chat, you asked, what's the longest reported survival of transplanted RGCs in a host retina? Does anyone happen to know the answer to that? What's the longest that anyone's ever looked? And then sort of a follow-up question there is, does anyone, what's known about axons transplanted in the eye, leaving the eye through the optic nerve head? How far do they get? I mean, I know there's quite a bit about, regenerating axons in sort of like an optic nerve crush model. But in terms of actually transplanted, I know I've seen, axons sort of making their way to the optic nerve head, but what about actually getting past it?
We did a squirrel monkey, published a squirrel monkey report from a human ESL derived neurons, retinal neurons transplanted into the squirrel monkey. We went 3 months. So 3 months for that. I don't know if people have done more than that in a mouse, but 3 months in a squirrel monkey was our.
Okay. Yeah, I'm just thinking because that survival period determines how much you can experiment with factors, trying to attract an axon, trying to guide it. Right? If currently you only have 7 days before the ganglion cell is dead, there's only so much you can do in each trial. And the thing has to start moving right away, or you won't know if it actually knows where to go. But 3 months is impressive, and that's a human cell in a primate, and they stayed there. You could tell viable based on fluorescence.
Yeah, axon growth was great intraretinally, not directed necessarily to the optic nerve head in that report. Some of them wandered around out into the periphery. Many of the axons, most of the axons, I would say, were not in the ganglion cell fiber layer, but they were in the plexiform layer. But they formed decent bundles, and they avoided the, they looked like the arcades going around the fovea. So some of the targeting stuff was still working in these cells, and they stuck around for 3 months.
Interesting. Thank you.
There's one monkey.
Yeah.
So
this is Tom. Sorry, this is Tom Johnson. Sorry to join late. I am in the car and just left another meeting. But we've been doing this in rhesus macaques, and we've done it in about 8 monkeys so far. We've seen RGC survival out to 4 months. And importantly, we see about the same amount of survival between 2 and 4 months. So it seems like there's a lot of attrition immediately after transplantation, but after a few weeks to a couple of months to let them settle down, we're not seeing a continued death that seems to be prolonged after the transplantation process. So it seems like there is some time window, certainly with surviving cells, to be able to experiment and look at ways of either improving survival or improving integration. And then in mice, we haven't done this in monkeys yet, with tissue clearing and having composite tissues where the eyeball is still connected to the nerve. We are now seeing axons within the proximal but retrobulbar and myelinated portion of the optic nerve. So we do think that they can not only home to the optic nerve head, but the axons do make that turn into the optic nerve. And we've only gone out to at most 4 weeks. So they're not very far along the optic nerve, but we're definitely, following them out now and seeing how long we can push them to see if they do get back to the brain.
Can I ask you?
And I came. Sorry, go ahead.
What's the origin of those ganglion cells then? Was that mouse ganglion cells?
Yeah, these are all human pluripotent stem cell derived RGCs. And we immunosuppress pretty aggressively because it's a xenograft. So in the monkeys, they get cyclosporin, methotrexate, and prednisone.
Yeah, but you're talking about mouse there in your cleared preparation.
In mouse, we use cyclosporin, or we transplant into NSG mice, which have no adaptive immune system.
Yeah. That's cool. Yeah. Thank you.
Yeah. I came on right as there was discussion about calcium imaging in donor RGC. So I kind of missed that. But if anyone has any lingering questions or thoughts, be happy to readdress that because we've done some work on that.
Yeah, I think some of the consensus was it's very exciting, and I was excited to hear about this, but we want to see the spikes. The calcium can be from a lot of things. It's not necessarily functional. It may or may not be. There's just a lot of, has anyone stuck an electrode on there and recorded light-driven spikes?
Yeah, no, I get it. I mean, they're definitely light evoked, both because it's temporally associated, and when we block synapse, when we block neurotransmitter release at the synapse, we can't detect light evoked calcium transients anymore. But they do have sort of a bizarre morphology, and we don't patch clamp in my lab, but we're working with some collaborators to get some true ephys whole cell stuff going on that.
I think the fact that they're synaptic is pretty neat. If you're putting on synaptic blockers and they go away, it says it's not, kind of just a melanopsin response, which is pretty cool.
Yeah, exactly. It's not. It's light evoked and it's synaptically mediated, but what it is on a true, are they action potentials? Is it some kind of bizarre complex? We don't know for sure yet.
Right. Yeah, Brad, go ahead, chime in.
Oh, I was going to loop back to the guidance question and relating to severity when we follow post-transplant eyes that have more or less end-stage glaucoma with very, very few intrinsic, host RGCs left. The patches of engrafted donor cells tend to send neurites toward each other. But when we transplant into eyes that have quite a bit of nerve fiber layer remaining, the neurites tend to grow along the existing bundle tracks, for what that's worth. So that seems kind of interesting. That's just in the retina, though, I should say, to the point that someone raised about myelination and being potentially a barrier to some of those signaling.
Yeah, so can I kind of follow up on that line of discussion about, if you want axons to follow their way through the nerve and the tract and, get to the LGN, and you have lingering axons that sort of are the road for regenerating axons to follow. What do you think is actually needed? What properties of those remaining axons need to be there? Do they need to be non-myelinated axons, or can they be myelinated? I don't know that kind of field and that world well enough, and so I'm curious if someone can chime in and share some insight there.
One of the major ways axons follow other axons is with L1, and L1 is a cell adhesion molecule that these axons specifically have, and so that's definitely going to be something that you want to keep exposed. At the optic nerve head, right, there's the of DCC and netrin. And so, you can imagine the other factor that gets axons to go past the optic nerve head in developmental studies is that it switches from using DCC, which is an attractant to netrin, a receptor that'll be attractant to netrin, and then switches on like Descam or one of the UNCs or that to then move past the optic nerve head. So it seems to me that if you did some combination of ensuring that there wasn't a attractant receptor anymore once they get out of the optic nerve head, and then that these cells would switch from, and potentially these transplanted cells are on a clock and they may switch from, once they hit their netrin, switch to an UNC or Descam and then move on past the optic nerve head. So ideally, that would be what you'd want, and then have L1, somehow on the remaining axons. But I think L1 sticks around. We've done, you can stain for L1 immunostain, in adults, and plenty of it on the axons. But again, the myelin accessibility is an interesting question, and I wonder in glaucoma, is there some loss of myelin that might actually, before the axons are gone, that might actually be a benefit for?
There is, but only just behind the lamina cribrosa for micrometers. I mean, some people are extremely, deeply interested in that question, but it's a pretty limited spatial phenomenon.
Yeah, glaucoma is not a demyelinated disease, but the key at the nerve head is that when you have lost tons of axons, the holes, the pores that used to go through where the axons need to get their way out are filled in with astrocytes that produce additional collagen 4 and even fibrous collagen. So much as Tom figured out you had to get rid of the ILM in order to get the cells to get into the retina, we may need to get rid of that blocking material to some extent, because otherwise the axons are going to run into some very inimical tissue blocks.
Yeah, we've recently looked for netrin in the optic nerve. I mean, it's obviously in the optic nerve astrocytes during development. You see it nicely there, but from some of the data, the single cell data sets that have been published, and you don't see netrin in the astrocytes in the adults. So I suspect that the ones that would be beneficial at the optic nerve head are gone. And as you're saying, the ones that are proliferating there, perhaps they're just not going to be that conducive for.
Well, they still got the original program. They just have changed their phenotype. And much like these stem cells that Tom's putting in, they seem to understand that they shouldn't send their dendrites out of the inner plexiform layer too far. A lot of, I mean, Tom, you can see the pictures in Tom's work, and you're depending on them to know what they should be doing. And one of the other speakers here was talking about exactly that.
So we've actually seen netrin in the optic nerve after a nerve crush. It's upregulated immediately after injury, and it's in the OPCs.
It's in the OPCs, did you say? Yeah,
throughout
that's where it
the
shows
nerve.
up. That's where it shows up on the single cell data in adult monkey human.
It's present in normal adult nerves, but it's upregulated after injury transiently. But the problem is DCC, and that we kind of stalled on that for technical reasons, but happy to chat more about that offline. But my question is, and I think Harry brought this up, further down in the optic nerve in glaucoma, has anybody looked at whether there is demyelination? Because I know there is it's sort of a time course, right? There's axon transport defects, and then there's degeneration of the axons. But somewhere between that, it becomes demyelinated first before these axons degenerate.
I'm sorry, you broke up a little bit. I wonder if you can repeat what your question was.
Oh, yeah. Further down in the optic nerve, do we know whether between axon transport defects and axon degeneration, whether these axons become demyelinated?
They do not in the optic nerve per se. As Brad said, there's some evidence from the Oregon group and Brad and Claude Burgoyne that maybe there's some demyelination right behind the nerve, but there's an issue there because the whole nerve head is moving posteriorward by, say, several hundred microns. And are they the same axon areas that are being measured before and after that whole movement goes? But do we really need myelin? Maybe, as Tom says, you need it for guidance, but I'd be perfectly happy with a whole bunch of new axons that didn't have any myelin, and so they're transporting a little more slowly, but the person might very well be able to see, just as the multiple sclerosis patients aren't normal, but they're still 20/30, 20/40.
Can I ask a question about the vasculature in these patients? So if we're thinking that the newly regenerated axons are meant to follow the tracks of surviving ones that stay resilient regardless of the injury, do we know that the vasculature in these patients is also relatively intact to facilitate proper guide or response to guidance cues, whatever those are? Or is that a pathology in and of itself that's intervening as a blockage?
It's not even remotely intact. The inner retinal vasculature is completely rarefied in end-stage glaucoma. It's gone, in other words.
Yeah, he's not correct about that. And Brad and I have a disagreement, I'm sure. I've
In the
actually
nerve head
found
you published.
So I have both monkey and human data that shows that there are many remaining capillaries, but there's a decrease in the capillary vasculature in exact proportion to the loss of neural tissue.
In
But
the nerve head, I agree with you, Harry. Absolutely. That 1984 paper of yours,
Yeah.
our in vivo data say the same. But in the retina, the capillaries of the inner, the RPCs are, dramatically reduced.
I think we're in agreement. They're dramatically reduced. And
Yeah.
yet, if you do a fluorescein angiogram, there's still blood flowing there. So
Oh, yeah.
we're gonna have to be hoping that there's sufficient for 10,000 new ganglion cells to survive in the macular area. And maybe because everything is so much thinner, they're going to pick up some blood from the choroidal circulation even. But your question is correct. Is there good blood flow all the way through the retrobulbar optic nerve and chiasm?
Mm.
And if you look in the nerve head or the nerve, there are about 2 or 3% of the volume of the tissue is capillaries, but the volume of tissue dropped, but the number of capillaries is still 2%, which means it downregulates dramatically to take account of the fact that the capillaries would be too close together. So they structurally autoregulate.
Understood. I'm also just thinking though, if even before the axon exits the optic nerve head and has to deal with any issues pertaining to the tissue after that, I'm thinking even within the retina itself, if you have surviving transplanted ganglion cells that now have to extend an axon and follow some sort of cue, how does the destructed vasculature that they are in contact with interfere with their ability to even respond to cues that may or may not be present or may not be introduced by us is what I'm trying to say. And if is it something that we can address by correcting the vasculature defect, or is it something that has to be overcome in the transplanted RGCs that have to be maybe engineered better, is what I'm thinking.
Well, comment, but the bipolar cells are all still there in advanced glaucoma. Glaucoma eye that's no perception of light still has neurons that are normally supplied by the retinal blood flow, by the retinal circulation. So while Brad's correct that the innermost layer, now seen in optical coherence tomography, angiography, next to the ganglion cells and nerve fiber layer is dramatically less than it would be, why wouldn't the bipolars all die if that retinal capillary circulation wasn't still intact deeper to them? And of course, neovascularization is rampant in various retinal diseases because new blood vessels are called forth by signals. Would our new ganglion cells send out a signal to give me a capillary?
Yeah, I think these are all reasonable points, but just to return to the light response thing again, what kind of light responses have we seen? I'm looking at this. Yeah, look at this paper. It looks like cobalt blocks them. Have you guys tried AMPA NMDA block? Do we know what receptors are doing this? Are they all 10 seconds long in the light responses, or are there anything that look like potentially more normal light responses? I don't know.
Is that a
So
question to Tom Johnson?
Yeah, we're
There.
doing experiments now to look at all different sorts of neurotransmitter receptor antagonists to see what properties of these synapses we can discern and what neurotransmitters are mediating this. The calcium fluxes are abnormal. They're long. They're not all 10 seconds long, but they're not the same as when we take an AAV and drive GCAMP expression in endogenous RGCs.
Right.
But to Brett's point, I mean, even sick RGCs start exhibiting bizarre calcium fluxes in response to light. So, they're responding to light through synaptic means, but what they're doing exactly
It's synaptic because of the cobalt block. I mean, that's going to block calcium channels, right? Isn't it gone because it's not necessarily just that it's synapses. Cobalt is blocking the calcium channel.
Yeah, but AMPA receptors also block it.
Oh, it does? Okay.
And when RGCs fire spontaneous action potentials on microelectrode arrays, the cobalt chloride does not block those. So I think some of the chloride intracellular is coming from endogenous stores in like the ER, not purely from membrane-bound calcium channel.
Yeah. No, I mean, it's exciting. Don't get me wrong, it's very exciting. It's just like
No, I mean, I
a lot
totally
more has to
agree.
be done.
Yeah, for sure.
Quick question. I don't know if this was already answered, but are they always on responses, or have you also seen off ganglion cells?
We have not seen off responses as of yet. I mean, the number of cells that we've profiled, though, is very small at this point. We've looked at probably 500 RGCs. Only a fraction of those respond to light, and then of those, all that we've seen so far have been on responses.
I see. Of course, to your defense, the on bipolar cell outputs are nearest to the ganglion cell layer.
Right. Yeah.
But that's intriguing too. Thank you.
I had a question following up on the vasculature line. So there's a nice paper from Shin Duan that shows that in a glaucoma model that the surviving RGCs tended to have be proximal to the vasculature. I was a question for the transplanters. When you transplant in ganglion cells, do the ones that stick around, are they in close proximity to the vasculature as well, or has that correlation been looked at before?
In case Tom is still driving or attending to the road, I can just comment on our own. I don't have stats for you, but there doesn't seem to be any relationship to existing vasculature, or really much relationship at all, other than to the breaks in the internal limiting membrane, something that Tom has also, his group has led the way in demonstrating this as an important barrier to engraftment. So we're doing different kinds of so-called internal limiting membrane peel approaches to breach the ILM, and wherever we peel, that's where you'll get engraftment. Outside there's very little engraftment. In fact, to the contrary, where there's intact ILM, we get retinal progenitors landing and proliferate. So we're seeing two different morphologies. Some of the progenitors that get through the isolation, they're still not post-mitotic, as the RGCs have already differentiated to that state. So in the donor cells that land in open retina, so to speak, where there's been a break in the ILM, they tend to continue to mature as ganglion cells, evidenced, I'll say cross-sectionally speaking, by different markers, RBPMS, et cetera, and to develop neurites, and eventually to start exhibiting action potentials on current injection. And so they're continuing to develop this neuronal phenotype, whereas the ones that land on ILM seem very, very happy, if they're progenitors anyway, so they're CHEX10 positive, they'll be Ki67 positive too, so actively proliferating. And they form a bit of a membrane that's bizarrely very loosely adherent to the ILM. Like you can just, in pathology, you can just peel it off. So it's so loosely adherent, I was amazed that it stayed on at all. We can see it there in the OCT. It's just that it's just barely adherent at all, at least after fixation. And
do you think that's like very localized signaling, or do you think it's like the neurons that got deposited on top of the ILM, they just don't survive or they don't make it?
Yes, I think both. Right. Maybe not signaling per se, although there's probably some of that too, but just speculating, I would say the ILM being a basement membrane is just really a fantastic substrate upon which the proliferating cells are happy to grow, whereas neurons want to be in retina, not on ILM.
Right.
So
Brad,
Yeah,
Thanks,
how
Brad.
long do your,
I think we
how long do the ganglion cells last? What's your record for ganglion
we,
cells?
we've never gone longer than 10 weeks so far, but Tom said he's gone to 4 months in primate.
4 months, yeah. I'm just curious how general that is, because you're doing macaque as well, right? And
Yes, that's right. And with this current cohort that's transplanted now we're targeting for a minimum of 4 months.
cool. Interesting.
Thanks, Brad. I agree and have observations that mirror basically everything you said. The only thing I would add to the question about preferential survival neurovasculature, I agree. We haven't seen evidence for that. And Stella Mary, who's a fantastic postdoc at my lab, has been doing longitudinal adaptive optics ophthalmoscopy in mice, and actually looking at the long-term survival of RGCs, looking at the same RGCs within individual animals over time. And one of the variables she's been looking at is their proximity to blood vessels and whether that influences their survival. And it really doesn't seem to matter if they're directly adjacent or far from major blood vessels. They seem to survive at the same rate.
Just to add a point to that I just put in the chat, something to consider is that death isn't necessarily the endpoint to be looking for. In lots of metabolic situations, synaptic transfer goes first. It's way more energetic. Spiking can go first. So the cells could be very quiet and still alive. I've been doing a lot of metabolism oxygen stuff, and the folks in Utah know this as well, right? Franz Wittenberg has shown a bunch of this. Cells can be alive and very malnourished with a very small amount of oxygen. So just again, back to the ephys, we kind of need to see if they're spiking.
Just along those questions, instead of the vasculature, how about connectivity to mular glia? Is there a way to see that the engrafted ones are making more contacts to the mular glia? Because then that could be indirectly how they connect to the vasculature.
Yeah, we've shown that there's definitely some kind of contact with Mueller glia, and we came upon it because we were looking for intracellular material transfer as a potential artifact that could have led us to looking at, for instance, TD tomato cells in the host retina and assuming they were donor derived when they were host derived. This is something that's been a problem in the photoreceptor transplantation literature. And we did not find any evidence for transfer of donor derived molecules from RGCs to RGCs. But we did find a lot of instances where TD tomato and other proteins and potentially RNA is transferred from donor RGCs to host Mueller glia. We had a paper on that in Stem Cell Reports a couple of years ago.
Elizabeth, are there actual identifiable connections of Mueller glia to ganglion cells, or are we talking about transfer extracellularly via receptors in the membranes?
Well, I'm thinking more about the new paper about seroblock SEM, where they actually had to go there and see the fine connections. We're trying to label them with different like stochastic labeling approaches, kind of like Jeremy Kay's paper. Marla Feller has some of the labeling of the Mueller glia and then tracking them across in vivo like that. But I was curious to see if in the transplanted you would be able to see that.
We had published quite a long time ago now in the early 2000s. Lin Wang was the lead author, doing some EM, not scanning block-face EM, but EM of human and non-human primate nerve fiber layer. The paper is ostensibly about varicosities of ganglion cell axons, but interestingly, there were numerous examples of like hemidesmosome, like electron dense sort of hemijunctions between RGC axons and Mueller cell processes in the nerve fiber layer. And it looks like there's a comment here from Seth Blackshaw that this has been shown. It looks like in development. I should click on this link.
Yeah, the focus of this paper is on IPRGCs, but the data suggests this is happening with all RGC subtypes, or at least a large fraction of them.
Yeah, I think, thanks, Seth, for that. I think that's, of course, an important thing to keep in mind that, like, yeah, this particular paper was in development, but in a transplanted cell or a damaged cell or a damaged retina, it's not at all clear if we could have gap junctions, nanotubes. Like, there's a lot of ways for which in which cells can connect to each other. And Mueller glia are absolutely gap junction strongly to certain amacrine cells already. So it wouldn't be shocking if they're all, I've seen parasites connected to other amacrine cells. Like there's potential for a lot of transfer.
It actually seems to be gap junction independent and mediated by
in this one.
neurotransmission. So that's an extra twist. But
yeah, but any of those could happen. Yeah.
All right, I want to be conscious of our time. We have about a couple minutes left in the scheduled time, so if there's any other questions, feel free to chime in now.
Oh, there was just one other thing, sort of off this whole, back onto the optic nerve and optic tract question. So wondering if anyone knows about physical barriers there and whether a detailed EM study would be interesting. I've just been talking with some collaborators about some possibilities there and whether there's anything interesting. It seems like, again, I'm an outsider, but it seems like there's quite a bit of molecular work there, and the field is doing a great job at identifying some of the important molecules. But in certain diseases, if there's particular physical barriers, would there be things that EM or some other imaging modality would help us know or try to clear out of the way?
Sorry, were you talking about in the nerve itself or at the nerve head? I might have missed that.
Yeah, more in the nerve itself, probably. Well, or the nerve head. I mean, I guess it depends on the disease and it depends where the thing is damaged. But like, yeah, maybe sometimes the axon regrowth is just stopped at a point because there's a scar there and because there's stuff that it can't grow through no matter what molecules you put in there.
There's an awful lot of histological study of both human and monkey optic nerve after glaucoma monkey model as well as human tissue. And I was mentioning there's extracellular matrix production by the astrocytes. Oligos are largely gone as far as we can tell or identify them.
Okay, but the ECM you think actually creates some barrier that makes it tough for the axons to grow through?
Don't know. I mean, it's not there in embryologic life. So when they first grew, they didn't have to grow through a lot of connective tissue because they were there first. They were going through primeval goo.
Yeah. No, I do. It's just that, right. Yeah, I just wonder. I mean, it's sort of well beyond me, and I could get you in touch with or someone who, if they're interested, in touch with Bobby Kasturi and some of those folks over at U Chicago, because the optic nerve is one of and optic tract are some of the tissues they've been looking at for their high-speed EM stuff. And it's just a situation where I don't know. I don't know if you can see enough that way, but especially with particular axons labeled in an EM way that you can actually see them, it might be kind of interesting to see like the physical structure there and whether there's anything we can learn.
I mean, if you just look at astrocytes, there is a big aggregation of astrocytes, right? After injury, you stay there for a while. It's as far as whether there's CSPGs, etc. I don't know, but my sense is that it's nowhere as severe as the scar that forms after like a spinal cord injury. So in that way, it doesn't form a cavity that physically prevents growth through. It obviously looks like these axons are able to get through the scar site.
Yeah, that I was going to just say exactly the same thing. The spinal cord injury people spend a lot of time worried about the astrocytic scar and the cavitation, but that's never been as big an issue in the optic nerve. But I think there's evidence that there's astrocytic proliferation after, say, axotomy, a nerve crush or something like that. So, which, it classically people have thought that the scar that is present in the mammalian optic nerve formed by astrocytes would be less permissive than, say, in a fish or a frog where there's no such scar tissue formation. But
there's definitely astrocyte proliferation by Ki-67 in mouse glaucoma models, and Lizzie Kimball, was on, and she's not still, has quantified that. I would love the group the next time to discuss whether there's going to be anybody to talk to when the axon gets to the central brain site, because their transsynaptic degeneration may be another issue that we have to deal with, and almost surely we have to.
Yeah, I'll just say my lab works on mouse DLGN, and we find that at least the relay neurons are still there. They have some somatic atrophy, some dendritic atrophy, but I think probably as you get further and further along, you're going to start losing some of those DLGN neurons, and then even they might be losing some of their connections to the cortex.
There's both monkey and human data on that.
Yeah.
Yeah, we've looked up to 8 weeks after it's a partial lesion, so it's not as severe, but up to 8 weeks, and the targets aren't losing anything yet, just not physiologically, just looking at markers.
All right. So since we are at the end of our time together, just want to say thank you all for coming. Thank you to our speakers for kind of seeding the discussion and helping lead some of the discussion. I want to say if you have feedback on this event, if you want to volunteer to help participate in a future virtual discussion, please send me an email. I'm the one who's been sending out all those emails to the RReSTORe email list, so you can find my email address there. And I think that's all I had to say about that. Thank you all for your time and for tuning in. And again, I will post this, or we'll get this posted on the RReSTORe website as soon as possible. All right. That's it. Thank you all. Have a wonderful afternoon.
Thanks for leading this, Matt. Great discussion.
Thanks, Matt.